So in many ways, the eye is nature's camera, and it has a lot of the same parts. So if we take this model of an eye, which is slightly larger than scale, and we open it up, it's got a lot of the same parts. So you have a lens, a series of lenses actually, that focus light onto the eye. So you have the cornea, which provides most of the magnification, and then the lens of the eye, which is an adjustable lens, which can stretch and change shape to focus light by the action of specialized muscles called ciliary muscles. So the light's then focused back onto what in a camera would be a sensor, but here is the retina, a specialized tissue in the back of the eye. This transduces light energy into the activity of neurons. And all this stuff in the middle, the vitreous and the aqueous humors, is basically to allow the light to pass through and transmit. So just like a camera, the eye can also adjust the amount of illumination entering into the camera. So a camera has an aperture, which you can open and close, and the eye has a pupil, which isn't so much a structure as an anti-structure. It's an opening, the size of which is controlled by the iris, which can open and close to let in more and less light, which controls how much illumination reaches the eye. It also controls the depth of field, how shallow the focus of the eye is. Now, just like a camera, the eye also has a number of other interesting features. The eye can pivot and move around, controlled by the ocular motor muscles, which control the azimuth rotation and the elevation rotation. There's also muscles that control the torsional rotation of the eye. So when we tilt our heads side to side, our eyes actually counter-rotate to try and keep the image stable. This is sort of like a pan head on a tripod to control the eye. In addition, the eye has a number of other nice features, like the ability to clean itself. So it has glands that can produce tears, and then blinking action, which can remove dust and dirt from the eyes. The eye also has a couple of fancy features that allow it to protect itself and repair itself. So if you have modest damage to your cornea, in general, your eye can repair that. So just the same way that a camera has electronics in it, the retina is actually quite a bit more than just an image sensor. So in addition to sensing the image, it also processes it and then sends it along the cable, the optic nerve, through the back of the eye. And this transmits the signals to the brain. So let's take a tour of where information goes once it reaches the brain. So I'm going to take this brain, I'm going to set it down here for reference. So information starts at the eye, light entering and being transduced by the retina. And from the eye, it goes along the optic nerve to a way station in the middle of the brain, between the cerebrum and the midbrain. It actually would be inside of this hemisphere of the brain, called the lateral geniculate nucleus of the thalamus. And from there, it goes all the way to the back of the brain to cortex. Now, just to orient you to some geographic terms that we use for anatomy, this direction we call dorsal. And an easy way to remember this is if you've heard of the dorsal fin of a shark or a whale, that's the same kind of terminology. And then this direction we call ventral. This direction we call anterior, meaning front, and this direction we call posterior, or towards the back. Now, once information leaves the eyes, it goes along two nerves called the optic nerves. They cross at a region called the optic chiasm, which happens near the base of the brain. And then from there, information goes to a way station called the lateral geniculate nucleus, which is in the thalamus, in the beginning of the brain, between the midbrain and the cerebral cortex. From the lateral geniculate nucleus, information goes all the way to the back of the brain. So even though the eyes are in the front, the information goes first all the way to the back, to visual cortex, which is an occipital lobe, which is in the back of the brain. So from occipital lobe and primary visual cortex, the first area of visual cortex, the information actually goes in a couple different directions at once. So we say that visual information is processed both sequentially and in parallel. And what we mean by that is the information flow is a bit like an assembly line, or a series of interconnected assembly lines. So visual information first arrives in area V1, and then there's one pathway, a succession of visual areas that go down along the ventral surface of the brain. And then there's another pathway, a series of sequential areas that go up the dorsal side of the brain. And we call these the ventral and the dorsal pathways. Now, most things in the brain are characterized by a huge number of exceptions. And even though the dominant flow is thought to be in this sort of sequential pathway-like organization, there's actually quite a bit of crosstalk between these two pathways. So parts of the dorsal pathway will connect to parts of the ventral pathway. But these dominant processing pathways have some really interesting features, which is why neuroscientists who study vision are very interested in them. So let's take a brief tour of the changes that occur along one of these pathways in visual cortex. So let's start with the ventral visual pathway. We start in the occipital lobe at the back of the brain in primary visual cortex, area V1. Then we go to an area called V2, which is just in front of it. And then we go down along the ventral surface of the brain to an area called V4, and then an area in primates called inferior temporal cortex. And in humans, this is roughly speaking sort of on the bottom of the brain. And we draw it with these arrows because this is a processing hierarchy. Information goes to V1, it's processed, it's passed on to area V2, it's processed and passed on to area V4, and so on. Now, a number of features change as we move from one visual area to another. So, for one, if we look at the receptive field size, we move from small receptive fields in area V1. So any given V1 cell will only respond to stimulation in a very small region of the visual space, to progressively larger receptive fields in V2, larger receptive fields in V4, and then sometimes very large receptive fields in area IT. So you'll find cells in IT that will respond to stimulation almost anywhere in the receptive field, maybe a whole half visual field. So that's one progression that we find along one of these processing pathways. Another that we find is a progression in complexity of the stimuli. So what do I mean by that? So in area V1, we find cells that seem to respond to local orientation, local edge structure. So maybe which way is a little line oriented in a very small region of visual space. And then as we progress along the ventral visual pathway to area V2 and area V4, we find sensitivity to longer and longer features, so perhaps integrating contours in the case of V2. And then we also find selectivity for different things like curvature, and we start finding cells that respond selectively for corners. So you can start to imagine that what's happening here is we're building up progressively bigger and progressively more complex stimuli representations as we move through the ventral visual pathway. And then by the time we get to the area IT, we start finding cells that will respond selectively to very complex objects, whole objects in some cases. So some parts of inferior temporal cortex in humans and primates seem to be selectively built to process faces. So you might find cells that will only respond when they look at faces, or perhaps particular faces or particular views of particular faces. So you might start to imagine that what's happening here is we're building up a representation of what's in the visual world. And because it seems to be concerned with what's in the visual world, oftentimes we'll call this ventral visual pathway the what pathway. This is all in contrast to the dorsal pathway, which seems to be much more concerned with where things are and perhaps also how we should interact with them. So the dorsal pathway includes areas with names like MT for middle temporal area, which seems to be concerned with the direction of movement in a visual field. So if an object is moving through its visual field, visual receptive field, it'll respond more, and they're tuned for direction of motion and velocity of motion. We also find areas like the posterior parietal area, which seem to be concerned with directing attention to particular parts of visual space. So you might have an intuitive sense that when you look out at the world, you don't pay attention to all of it equally all the time. And in neuroscience, we call this spatial attention. So we can focus our attention on one particular region and really concern ourselves with what's happening there. And it's thought that parietal cortex and the dorsal part of the brain, the visual cortex, seems to be involved in that kind of processing. So you might wonder how we know all of these facts about these different pathways. Well, one way we can do that is to use the same sorts of approaches that Hubel and Wiesel did and put electrodes into the brains of animals and see what those cells do. We can put stimuli up on the screen, and we can see how those cells respond, and we can characterize them. But another interesting way we can do that is we can look at what happens when different areas are damaged.